Fantastic. Thank you. Thank you, Jean and Vince, for inviting me to be here. All right, now I get to find out which of these buttons does which.
Right button is forward, left button is back, the arrows.
That's what I thought. In the event, my arrows, oh, there we go, I made it. Okay, fantastic. So I've been talking about big data from, in the imaging genetics as a big data problem for a few years now. Whether you're dealing with common variants, so it's very easy to find people with genetic variation at a particular genetic loci, or individuals with rare variants, where you might expect the effects on the brain to be larger, but it takes a lot larger sample to actually find anybody with a genetic variant, you're going to need massive data sets to find just these associations if you're going to look across the entire genome. As has been indicated already in the talks previous to this, there are a lot of steps between the genome and eventual brain measurements and symptoms and disorders that we're all really interested in. And of course, finding this relationship, just finding the association, is really only the first step, not the last. But let's see what we can do in order to get to the first step. So I'm going to talk about the Enigma project, or Enhancing Neuroimaging Genetics Through Meta-Analysis. There was an entire workshop on imaging genetics that was largely populated by Enigma speakers yesterday. If you missed it, I strongly recommend getting it through on demand in a few weeks when it comes out, because it's in much more detail than what I'm going to talk about today. But it was started by Paul Thompson and a few others back in 2009, 2010 or so. And the joy of the approach, from my point of view, the strength of the approach, is really everybody who has relevant data gets to play, right? Everybody can join in. It's not limited to just the PIs who can wangle a huge amount of money out of a funding source in order to do a very careful prospective analysis that requires a great deal of standardization and huge populations in one particular region. But it really does depend on lots and lots of groups that may have smaller samples, 200 subjects, maybe even 25 subjects, but they have the relevant data and they're willing to play together, and we can actually do some analyses through using meta-analysis, basically looking at each individual group. No one group is sufficiently powered, but can we look for a consensus effect across multiple studies and see where the strength of the evidence is really pointing us? So many of the pictures and graphs and everything I'm going to be talking about today were also pulled from this recent paper about the Enigma Consortium that Carrie Bearden and Paul Thompson published, I think, last year, 2017. So again, that's another resource for you for more information. I'm one of the co-chairs of the Enigma Schizophrenia Working Group. I've been running that since 2012 with Theo van Erp. And what we've been focusing on is what we could call prospective meta-analysis, as compared to a standard meta-analysis where you look at the results that have been published from the literature, regardless of how they were analyzed and how they were done, and you look for a consensus effect. What we're doing here is saying, look, we know everybody collected their data differently, right? Some of this data was collected in 2003, some of this was collected in 2010, whatever, and it was all done for their own purposes and for their own studies. But what we can do with the Enigma approach is say we're going to standardize what we can and try to model the rest. So we standardize what everyone does with their imaging data in order to extract the brain measurements, and we standardize what everyone does in terms of the analysis so that the meta-analysis is then that much cleaner. The data are not aggregated. When the first papers came out, people often came to me saying, oh, can I get the data? Can I get the data? And I'm like, no, you can't have the data. The data is sitting at the same sites that collected it. I don't have it. What we get is results shared from everybody. Everybody does the analysis and shares the results. And I'm going to put a asterisk by that because, of course, some groups have decided that they do want to aggregate their data in order to facilitate certain kinds of analyses, but many groups cannot. And so we work in a meta-analytic framework. This is sort of the baby picture of Enigma from the 2012 first in imaging genetics paper. These are the contributing sites, largely North America and Western Europe, but some international sites. Again, The focus was on standardizing, not data collection, not trying to make sure the imaging protocols are the same or that the genetic processing is the same, but that the post-processing is the same, so that you've done the same imaging analysis to extract the volumes, you've done the same quality assurance for throwing out people for the same reason across sites, and we have standardized statistical models, all the covariates are treated exactly the same way, et cetera. And the genetics were the same. The imputation, very carefully controlled, the QA steps very carefully prescribed, and standardized statistical models. So there we go. There have been sort of three major progress milestones, if you will, for the Enigma imaging genetics. The first one in 2012 is just, let's start at the bottom, start with hippocampal volume. Everybody wants to know about the hippocampus, and an intracranial volume. And then Enigma 2 in 2015 published the subcortical, looking at all the subcortical regions. And then Enigma 3, which is currently ongoing and hopefully going to be a paper very soon, is really looking globally at cortical area and thickness measurements using this full genome-wide association study. And I want to really point you to Neda Jahanshad's presentation Thursday morning, where you'll get all the details about what Enigma 3 is finding out. This is an example from the Enigma 2 paper. It's out publicly available from 2015, using about 30,000 subjects from 50 different cohorts, 300 co-authors. It's a mammoth kind of thing, but we were able to look at the entire, well, not the entire, but the genome-wide association studies to say where the associations are with the hippocampus, the putamen, et cetera, replicating the previous hippocampal results and extending it into new subcortical areas, and of course what that all means. The way this is actually done within Enigma is you're not just one big consortium, right? We're a loose family of working groups, and within each working group we have multiple sites that are participating. So I started with the schizophrenia group. There, of course, right after that came bipolar. At the same time came bipolar. Shortly after that, major depression groups. And those groups are all largely interrelated. We have a lot of overlapping collaborations and interests and interests in comparing results. There are, since then, we've spawned off an early onset psychosis group, and new groups are coming up all the time. We just had a meeting to start about clinical high-risk groups, things like that, anxiety disorders, ADHD, all of the disorders. There's now up to 22 clinical disorders working groups and new ones showing up again all the time. But something that we can do now that we have this worldwide framework is to start really leveraging the rare genetic disorders. So Carrie Bearden's running the 22q deletion syndrome working group, which again, it's difficult to find, but incredibly relevant to many psychiatric disorders. But if you can work in a worldwide framework, then you can actually identify enough people with this kind of a deletion syndrome to really say some important things. So we've all published our initial imaging papers looking at the largest meta-analyses of subcortical volumes, cortical volumes. Those have been out for a few years now. We can start looking across the disorders to say, how are the patterns of cortical thickness and subcortical differences in volumes similar or different across the different disorders? And what does that mean in terms of diagnostic boundaries? And the Enigma 22q got their first paper out, I think literally in the last few weeks, looking at cortical effects with sufficient power to really say what's going on there. There are within the Enigma umbrella non-clinical working groups that are looking, for example, at just techniques. How do we develop the harmonized pre-processing techniques for DTI, resting state, EEG, sulcal morphometry, shape analyses? How do you do that in this decentralized kind of federated environment? There are groups looking at non-clinical issues in brain changes, such as healthy variation, aging over the lifespan, without bothering with the disorders per se. And of course, there's the genetics groups. The genome-wide association studies was really the core that kicked off Enigma from the beginning. But within this framework, we can now start looking at the CNVs, at the epigenetics like Jean was talking about, and evolutionary genetics approaches. And again, these are all just groups of people who want to work together in many cases. Some of them have managed to get funding to support their particular group, but many of them, it's just, if you want to talk to us, come talk to us. It's not closed in any way. Another approach, besides merely being able to get the large data sets and the specific genotypic kinds of approaches, is to look at heritability. So one of the approaches that has been used multiple times is to say, if you have, again, a group here and a group there and a group somewhere else, all of whom have done their family studies and can look at linkage and can look at heritability. Now, if you can get them all to do the same image processing pipeline, you can see whether the heritability effects replicate across these different groups. And that is actually one of the ways that a number of the methodological groups are using to sort of vet their pipelines, to say, if we can reproduce the heritability in these different large multi-family, multi-generation studies, then we're happy that this is a robust pipeline we can roll out generically to say to these different groups. So this is the current status of the Enigma consortium worldwide. Paul Thompson gave me this slide from, I think, SOBP just in May. It's basically showing where we are in terms of participating consortiums. As you can compare it to the picture from 2012, we're now up to 63,000 samples from 39 countries, and again, 22 different brain disorders. Still heavily weighted towards North America and Western Europe, but we've really expanded out into the rest of the world as well to be a bit more representative, both for imaging measurements and imaging genetics together. And I want to just give you a teaser about what the Enigma 3 has been finding in terms of surface area and genetic effects. And of course, Neda will give you much more details. But yes, there are some genome-wide associated associations in the cortical surface areas in some very interesting regions, in this case, pre-central and post-central surface area. Now, I want to put some caveats in there. We did think, of course, right after the 2012 and 2015 papers, if you have the results from Enigma where you can say, you know, this across the genome and across all the genetic markers that we're working with, we know what, if you will, the risk for a different size of the subcortical volume, any of the subcortical volumes. We know what the risk is, for example, for the hippocampus, what the effect is on the downstream hippocampal side. In the middle, we know what the risk across the genome is for schizophrenia. So for the same markers, we know both how they're related to hippocampal volume and we know how they're related to schizophrenia. And from on the right is our Enigma schizophrenia meta-analysis, where we can sort of nail down what we knew already, that the hippocampus is the subcortical region with the largest effect size and the most consistent effect size. If you look at case control differences, we should see within the genetic risk for hippocampal volume where the risk markers are for schizophrenia, and vice versa. Within the risk markers for schizophrenia, where are the genes for hippocampal volume? And there's none. And unfortunately, this is an incredibly bizarre paper to work on, because no matter how you looked at it, there's no enrichment within the genes for the hippocampal volume for schizophrenia and vice versa. And this is the subject of an entire another talk of what does this mean for imaging genetics. But I just want to throw this out there as a caveat that we have to think carefully about association and what it means and what we're talking about in terms of our disorders. But looking forward, these experiments take a huge amount of time. The meta-analytic approaches require interacting with many, many people. The schizophrenia group has 50 participating sites as of right now. And I have to communicate, and Theo has to communicate with all of them to say, okay, what are you doing? Are you doing it right? Let's double check this. Let's keep moving, and there's a lot of back and forth getting everything set up so that it all works. What if we could actually automate it and make it go faster? Ideally, much of this is just working with the same data over and over and over again. Could we just say, okay, put your data out there, put it somewhere, and tell us what's going on, and then just tell us whether you want it used in a given analysis or not, and we don't have to bother you anymore, right? But how do you do that and still protect the subject's privacy so that nobody can reconstruct who they are? You want to make sure you get the credit, because these are incredibly expensive data sets to get. And what about the analyses that require really canonically that all the data be in one place, like a machine learning approach that usually starts with most of the data in one location in order to build it. So I want to tell you a little bit about the COINS Toolkit for Anonymous Computation, or COINStack, which is spearheaded by Vince. I'm on the sort of motivation and evaluation team for COINStack. The idea there, again, is you put the data out there, And you can actually say, this consortium is doing an analysis I want to participate. This consortium is doing an analysis, and I want to participate. We have a version working. This is an example just of a regression analysis. I know the text is very small, but it's basically saying, I don't know where the data are. One of the data sets is, one computer is on another computer. With this decentralized approach, the analysis can actually go do the analysis here, do the analysis there, and give us back globalized results without us ever having to see the data, see the images, or know what's happening there. And since this is VINCE, we know that there are lots of multivariate analyses on there. It's not just straightforward regression like we've been doing with the Enigma analyses, but actually lots of iterated approaches and ICA approaches, multivariate approaches, including a decentralized support vector machine approach. So there's lots going on there. And of course, I want to acknowledge the Enigma group on the left. Paul is buried in the back somewhere in his supporting role. The middle is my own imaging genetics lab, and on the right is Vince's medical informatics and analysis lab. Thank you.